We're going to the next presenter, Carlo Cardellini from the University of Perugia. He works mainly on diffuse degassing and deep CO2.
Okay, thank you. I have to leave. I have an appointment.
See you.
Okay.
See you, Lassie.
Okay, Lassi, thanks.
So thank you, Arthur. Now I'm trying to share the screen and start this presentation. Okay. It's okay?
Yes.
So good morning, everybody. I would like first to thank Arthur for giving this opportunity to do this such lectures or seminars. And I will talk about studying diffuse degassing from soil, and mainly I decided to focus on methodologies, and if there are time or depending on the time, we will see some application. And so the outline is that first we see some introduction and generalities about gas fluxes from soil, then measurement methods, and then approach it to that analysis. I think is another important part of the story. And then we will see how many applications we will be able to go through. So just to introduce briefly the idea, so in which geological environments we have that gas is produced at depth and is released at surface by diffuse degassing. Of course, volcanoes. In volcanoes, we have magma chambers and or geothermal system that can release carbon dioxide because it's both dissolved in the water, in the magma, or is dissolved, is present in the hydrothermal system. And so this gas, of course, can fit at the surface different type of emission, focused emission, but also diffuse emission, both even if it's directly coming from the magma or from the gassing of the hydrothermal system. So we can have situation like this. You see in this picture, you see the pointer when I'm moving.
Yes.
Thank you, Arthur. And you see we have areas like this. You see there is very scarce vegetation, but even where there is vegetation, we have a diffuse emission of gas from the soil, mainly carbon dioxide. For instance, this area is Solfatara of Pozzuoli is releasing about 1,500 tons of carbon dioxide per day. Of course, we have such emission, for example, also in geothermal system, even if it's not associated to active volcano. This is another case. This is an medium enthalpy geothermal system. We have carbon dioxide rich fluids in the system. This gas create a gas cap on the top of the system. There is some fracture, faults, and permeabilities on the cap rocks that allow the gas to be transferred to the surface. And you see here there is a picture of a field taken after a day of heavy rain, and you see bubbles in the stagnant waters. When there is no water, you cannot see anything because carbon dioxide is odorless, colorless. But this bubble is testifying this degassing from the soil. In fact, if you see on this side, we have what we want to produce, a map of the carbon dioxide degassing from the soil, and you see that all this area is characterized by high fluxes and the release of about 70 tons per day of carbon dioxide. But gas fluxes, carbon dioxide, but other fluxes, for example, methane, also characterize other geological system, other reservoir. This is, for instance, some images from an hydrocarbon reservoir. And also in this case, we have, together with bubbling, focused emission, we have area like this with diffuse emission of gases, in this case, mainly methane. But there are also some not geological, not natural systems that can be produced gas fluxes in a diffuse way and can be interesting to investigate, for example, for environmental application or monitoring or control that are, and these are the landfills. In landfills, we have bioreactor that is like our magma chamber producing gases, mainly carbon dioxide and methane, and these gases can move to the surface. And generally, for the regulation, this gas has to be not released to the atmosphere. So the landfill has to be controlled, But we can see, for example, from this map of the soil gas flux from soil, that you see the anomaly on the top of the cover of the landfill. So we will see that studying diffuse degassing can be also used not only in geological application, but also in some environmental one. But just to put a point, it's not trivial, it's not obvious that when we have a gas source at depth that we have soil diffuse degassing, because this is true at least for the soluble gas, like for example carbon dioxide, because the presence of aquifers of a large quantity of water between the source and the surface can completely hidden the carbon degassing from the soil, the carbon dioxide degassing from the soil. In this case, we need to use investigating the groundwater to detect the occurrence of these deep gas sources. But let's go to see what we want to learn. What are the main objectives studying carbon dioxide diffuse degassing, for example? You can have your own objective, but generally what we want to do is to characterize the source of the gas, to define the structures of soil degassing that, for example, allow to define the tectonic structure that control the release of the gas at the surface. So we can use the maps of diffuse degassing to trace, for example, the faults, or to identify the top of this gas accumulation zone. But another important objective is to quantify the total amount of carbon dioxide for different purposes that we will see also in some cases. And also another application of the study of diffuse degassing is to study the spatial and temporal variation of the degassing from the soil. That is generally valid, is used for monitoring purpose. The field of application are many. We can use this approach for volcano monitoring, for geothermal prospecting, for hydrocarbon prospecting, and for environmental studies. There is another field of application that now it's quite developing, or advanced, that is studying the possibility for carbon dioxide storage and sequestration, but also more general field of research, for example, the budget of carbon dioxide emission at global scale. And the first, we have to measure the gas flux. So how we can measure these gas fluxes from the soil? We can use some indirect methods or direct methods. The indirect methods consist essentially on measuring gas concentration along vertical profiles in the soil and using the equation describing the gas transport mechanism to estimate the flux from the measured concentration of the gas into the soil. But we will see that we need to assume or assume which is the gas transport mechanism. First, we need to know the soil property, and we need to know many variables. So at the end, the message would be, I think that it's better to, if possible, to go for a direct method in the measurements for measuring the gas fluxes. So to have an idea of the process, I think that we can see now to some basic principle of the mechanism transport in porous media. So we are going to see the process that is controlling gas flux, considering no interaction with the water in the soil or in the rocks. So depending on the source and in the permeabilities of the soil, the gas movement can be induced mainly by two types of forces. So it can be due to the existence of a concentration gradient, of a pressure, and of a pressure gradients. These two forces are going to control, to define two main transport mechanisms, that is the diffusion, that is driven by this concentration gradient, or the advection, that is driven by a pressure gradient. So when we can have in our system, in the soil, a zone with a high concentration of gas and the boundary condition of low concentration, the gas, that is this simple sketch, is represented by these colored balls, try to reorganize and so moves from areas where we have high concentration to low concentration. This mechanism, this type of flux, or the diffusion, It can be described by this quite simple law. So the flux is proportional to the concentration gradients. So from this, the idea that we can measure the concentration gradient into the soil, we can estimate the flux, but we need to know at least to know the values of the molecular diffusion coefficient that make this proportionality. But it is not quite simple, because we can consider different kind of diffusion coefficient. The simplest one is just considering the type of the gas. But if we have not only one species, but we have a mixture, we have to consider a binary diffusion coefficient. And if you want to take into account the effect of the structure of the soil or of the rocks, we need to consider an effective diffusion coefficient. So the system is becoming to become more complicated. But also, as I told you at the beginning, if we look, for example, the result of this experiment that's made on the lab, so we have this tank with sands, gas flux at the bottom. So we create a gas flux and we can measure the concentration. We can see that the concentration, depending on the magnitude of the flux, does not increase simply in a linear way as is predicted by the Fick's law. So that means that diffusion is not the only mechanism. In fact, we have this contribution from advection, and the advection is controlled by a pressure gradient. So when we have a gas with a certain concentration that moves with a certain velocity, we have a flux that we can estimate by the concentration of the gas multiplied by velocity. And it's this velocity that depends on the pressure gradient. So when we have a pressure gradient, we have a certain velocity. But again, this is depending on both the type of the gas, so the dynamic viscosity, but also from the structure of the soil, so the intrinsic permeability, which can be the source of this pressure gradient. Of course, we can have pressure gradient where we have localized gas generation, deep fluid reservoir, but also tectonic stress may induce variation in pressures, rock fracturing, but also an external parameter like changing in the barometric pressure. So the system is quite complicated, and also because diffusive and viscous flux are not independent, they coexist together. There are interactions between gases. Interaction between gases, and the structure of the media, and it's a multi-component gas flow. So the equations are not so simple as we have seen in the previous slides, and there are at least several models of very complex equations that can be used to investigate the gas transport mechanism, or for the reverse problem, to compute the flux from, for example, from measuring the concentration of gas at different depths. So the applicability of the different equation is quite complex. So just to don't lose too many time, my conclusion is that the gas flux from soil is preferable to be measured with the direct methods. Which direct methods we have? One of the methods is known like concentration method, dynamic concentration method, and in this case we are using a probe. We are sucking the gas and we are measuring the concentration of the gas with an infrared sensor. And according to the authors, the magnitude of the flux is proportional to the concentration of the gas after a certain time of measurements. So we can compute the flux on the basis of this concentration that is named dynamic concentration. But again, we can say to compute the flux, for example, using this equation proposed by Camarda and co-author in 2006, we need, if you want to be very accurate, we need to know the soil permeability. And the authors proposed a method that can be used to estimate point by point the soil permeability, but we need to use a gas generator, and we need to put air into the soil and to measure the pressure at different depths to estimate the permeability point by point. And sometimes this is not very useful for an extensive survey of gas flux from soil. The other method that is it's largely used in many applications is the accumulation chamber methods, and this method allows to measure the gas flux at the interface between the soil and the air. So we don't need to put nothing into the soil, maybe disturbing the permeability, changing the pressure gradient. So we are using like a pot that is placed on the top of the ground, and the gas, if there is a gas flux from soil, the gas is entering to the chamber and increase the concentration of the gas inside the chamber. So the gas is taken and measured by also in this case for carbon dioxide by an infrared detector, and then is reinjected into the chamber. These closed parts of the gas allows to don't alter the pressure inside the chamber. You have seen that the gas flux can be it can be induced by a differential pressure. So to do not disturb the real gas flux, to don't alter the natural phenomena, the gas is circulated continuously between the chamber and the detector. So we can read the concentration of the carbon dioxide variation during the time, and the slope, so the rate of the increase of the concentration is proportional to the flux. So in this figure, you see an example of measurement. So during the time, the concentration inside the chamber is increasing generally linearly, and the flux of gas from the soil is proportional to the rate of increase of the concentration by a geometric multiplied by a geometric factor, that is the 8 of the accumulation chamber. This factor, if you want to be more precise, can be determined, for example, by laboratory calibration of the instruments. We can use a tank of carbon dioxide. We measure the flux that we put into a very small soil, and we repeat several measurements of different known flux. So we can build this diagram that is representing the relation between the rate of increase of concentration and the flux. So we can estimate our equation that we can use for computing from the increasing concentration inside the chamber. We can compute the flux of the gas. This method is not only used for carbon dioxide and can be measured for all the gas for which we have a sensor that allowed to measure the gas and then to reinject the gas inside the chamber without destroying the gas. This is the example of the exercise that a researcher have to do on field. So you have our chamber, putting the chamber into the soil, taking the measurement, waiting for seconds. So it's also a very quick method that allowed to do survey in a not using very long time. So to talk about the applicability, so we can measure carbon dioxide flux with infrared spectrometry. Now there is also very nice instrument that allow to measure accurately also the methane flux, use lasers or infrared that is not very precise for the methane, but there is now this tunable laser diode based sensor that is very sensitive. There are sensors also for measuring volatile organic carbon, and some sensor for measuring also H2S. So this method can be applied to different gases that can be used in different type of investigation. This kind of methodology is also used quite frequently for the monitoring. This is an example of a monitoring station of carbon dioxide fluxes from by INGV in Naples. And practically inside this small house, there is an instrument similar to this. So we have our accumulation chamber that automatically can do the measurement without any researcher that live inside this small house taking measurements every hours or in the time that we want to measure. The same principle of the accumulation chamber method can be used also for gas for which we don't have sensor that can be used directly on the field for continuous measurement, because we can use the same approach in as is called a static way. So we place our chamber and we take samples of gas at different time, and then we bring back the samples into the lab, we do the analysis, and we see how much is increased the concentration into the time using several samples. So this method in principle allows to measure the flux of any gas species that can be measured in the lab. Of course, again, it's much more, we can say, expensive method in terms of cost, time, and instruments. So another important thing that is evolving in recent time is to coupling the analysis of the isotopic composition of the gas from the soil together with the measurement of the gas flux. We will see later in a few slides that sometimes it's not very simple to investigate the source of the gas only using the values of the flux, but we can couple these values of the flux together with the definition of the isotopic composition of the gas from the soil that can be much more effective in defining the source, the real source of the degassing. This is a method that was proposed by Cardellini and co-authors in 2008, and it consists in taking some gas, at least 2 gas samples during our measurement, and analyzing the isotopic composition of these 2 samples that are taken at different concentration during the measurement. We can estimate the isotopic composition of the carbon dioxide entering into the accumulation chamber. So we have seen something about the method that we can use, and now once we have measured the fluxes, what we can do with our data, we need to use some statistical and geostatistical tools for the characterization that can be useful for the characterization of the source of the gas, the definition of the spatial distribution and mapping of flux from soil, and can be used for quantify the total release of the gas. Here I'm talking about carbon dioxide, but this method can be used for all the gases. So one of the most used approaches is of statistical techniques for investigating and for the source of the gas and characterizing the gas are, for example, the probability plot. So the probability plot is a chart, a diagram that's allowed to plot our data and to define the main statistical properties of our data. And the particularity of this probability plot is that if we have a single Gaussian population of data, these are going to plot in a straight line in this probability plot. Take in mind this, because this will be useful to understand the next slides. But we can have that our data are not Gaussian distributed, but if we are lucky, working with the log of the values of the data, again, and using a log probability plot, our data can show this straight trend that is telling us that our data is representative of a single log normal population. But unfortunately, gas fluxes from soil, carbon dioxide fluxes from soil, as many other geochemical parameters, has no simple distribution. Here is reported the probability plot of the log of the gas fluxes taken from different surveys in different areas. And we see that just in one case, maybe we have this straight line suggesting that the data are referring to a single population. While in most of the case, we have this shape of the statistical distribution with some inflection points. And this complex statistical distribution generally are due to the mixtures of different statistical population of fluxes that can be due to the presence of different sources that are feeding the carbon dioxide degassing, or that can be related to different mechanism of transport or to large variation in the permeability of the soil. So what we want to do is try to decompose this complex distribution and to define the different statistical population that contribute to this complex distribution. This is one of the, I think, an easy case. So when we have, for example, one inflection point, we can model our statistical distribution with the combination of two populations. To make an example with carbon dioxide, we can have gas fluxes that is coming from the biological activities and from the soil that are mixed with gas fluxes deriving from the deep source. When we analyze the gas fluxes, we want to characterize both processes, because if we consider all the data together, we are characterize something that is nor the biologic flux or the deep contribution. In general, in a more general way, in these probability plots, the number of inflection point is suggesting us the number of population, because generally we have that if we have one inflection point, we have two population. If we have two inflection point, we have three populations. So the number of population is one more than the number of inflection point. So this diagram gives also to us other important information. For example, the probability, the cumulative probability of the inflection point suggests also the relative proportion between the population. So we can define the statistical parameter of the two populations, but also the relative proportion in the mixture of these two populations. So we have data on the relative proportion, so we can also estimate the average values of the single population and the standard deviation of the population. So if we have a complex data set with fluxes that is deriving from both from, for example, biological carbon dioxide production and deep carbon dioxide production, with this method we can say we can characterize both the biogenic components of the flux and the deep component of the flux. But for example, if we are in a situation like this, where we are working with the log of the carbon dioxide fluxes, so our data set is a combination of two log normal population, we have to take in mind that the average values and the standard deviation of the two populations that we define with this approach refers to the log of the values, not to the flux, but to the log of the flux. So we have to use another statistical method for estimating the mean and for example the standard deviation or the 90% confidence interval of the mean, starting from the mean of the log of the values. And for example, like suggested by the GSA method proposed by Cardellini and co-authors in 1998, we can use, for example, the Seychelles table, and that's we can estimate the mean values of the flux population starting from the average of the logarithm, but we have to consider also a correction factor, we can say, and this correction factor depends on the numbers of samples that are representing our population and on the variance of the sample. So this number may be very different from one. That means that's not neglecting this factor, we can do very large error in estimating the mean of the flux population without considering this correction factor. Because this is an estimation of the mean values, not an exact calculation, together we have also to compute the uncertainty of this estimation. And again, we using this Seychelles approach, there is another table that allowed us to estimate the lower confidence limit and the upper confidence limit for this average estimate. So before to see other things, I would like to present to you some frequent errors and misinterpretation of this diagram. So this is a picture taken from a book, and you see we have this shape of the distribution of the data. These are not fluxes, it's arsenic in soil, but it's the same problem. And you see the authors here are going to interpret and derive the different populations just fitting the different branches of this diagram. And this is not true. We have seen before, if we have an inflection point, or a situation similar to this, we have an inflection point here, the two populations are not simply the fitting of the values along the diagram. Otherwise, the interpretation of the data set that we have seen before, that is two populations, will produce the estimation of three populations. So if you want to use this method, please try to avoid this misinterpretation. This approach, at the end of this approach, what we have characterized the average flux, of the different population, and also the relative proportion of the different populations. So this method was also used to estimate the total gas release from an area. That can be done by multiplying the average flux of each population for the area that we think it's related to the different population. And we can compute this area considering the proportion of the different population and the total areas. For example, this is a table taken from a paper. So for each area, we can characterize the biological flux, for example, the hydrothermal flux. Knowing the proportion between the two populations, we can compute the total flux that is connected to the two populations. Note that this approach assumes that all the measurements are independent, so this is a pure statistical approach, but we need to take into consideration different possible limitations of this simple statistical approach. And one is that the partitioning of the population is not univocal and depends on the researcher that do this partitioning. But also, we have also taken into consideration that the use only of the parameters regarding the flux, the mean values of the flux to define the source, for example, as we have seen before, distinguish between biological and deep source, can lead to erroneous interpretation. And I want to give you an example taken from the study of the degassing of Solfatara of Pozzuoli that we have seen also in the first pictures. Here at Solfatara, this is data set from one survey of carbon dioxide. We see this very nice bimodal statistical distribution that can be interpreted with the combination of a low flux population and a high flux population. And this is the estimated mean for the two populations. So one population is 47 grams per square meters per day, and the other population is more than 2,000 grams. So it's quite clear that the high flux population is connected to a deep source. For the other population, we could be satisfied because generally 47 is the values of a low flux that can be also biological. But this area was investigated for monitoring purpose, and what was discovered is that that the values of the low flux population is going to change with time. So it started to arise a question: it's really this low flux population always due to the biological contribution? And because if you look to the different sources, we know that the carbon dioxide that can be produced by the soil due to the roots respiration and the composition of organic matter, that generally has values as I told you before, that is quite low. We have mean fluxes in terms of 7, 11 grams per meters, with maximum values that can be in terms of 30, 50, also 70, with variation that can be connected to different seasons of the years. So values of, as we have seen before, of 47 grams per square meters can also be in the range of the biological carbon dioxide. But we see that in the time at Solfatara, we had also very much higher values of the low flux population. So it's difficult to interpret this low flux population as completely linked to the biological flux. For this reason, was used the combined approach, including the measurement of the isotopic composition of the carbon dioxide. And if we look to the same probability plot done not for the soil fluxes, but for the isotopic composition of the gas released by the soil during the degassing, we see that here we cannot simply explain this statistical distribution with the combination of a biological and the hydrothermal components, but there is also, we need also an intermediate population that is something that is a mixture between the two. So the lecture, what it is, that's looking to the isotopic composition of the gas together with the investigation of the gas flux, was discovered the presence of two low flux population on the base of the isotopic composition. One, it can be considered the real biological background because it's characterized by low flux and by an isotopic composition that is compatible with the biological production. While the other low flux population is only apparently biogenic because this population from the isotopic point of view is not deriving from the only biological source. So we have a real background that is about 26 grams, that is very well in the range of the biological production. And we have an apparent background that is really a mixture of biological carbon dioxide and a low amount of deeply derived that represents just an apparent background. So in this case, the definition of the real background is particularly complex when because the deep fluxes can be also of low magnitude. So when we have low fluxes that are of deep origin, we cannot use only the interpretation of the flux values to define the source of the gas. In this case, we need to couple together the investigation of the flux together with investigation of the isotopic composition of the gas released. So now to do another part of the story, we have seen which method that we can use to investigate the source of the gas. But what we want to do is also to investigate the spatial distribution of the gas to characterize, for example, the geological structure that are controlling the degassing. And so to approach this problem, we have to use geostatistical approach that allows to characterize the spatial distribution of the fluxes, and that can be also used for the estimation of the total release of the gas, using an approach that is much more based on also on the spatial correlation between the measurement and not only to a pure statistical approach. For draw a map of the gas from the soil, we can use at least two different approaches, and that is estimation and simulation. And of these two big classes of statistical approach, both of these two big classes have some code software that can be used. For example, the most frequently used geostatistical estimator is the Kriging, or the most used geostatistical simulation is, for example, the sequential simulation, Gaussian simulation. Both methods can be used for mapping our distribution of fluxes into the space. They have different targets, they have different application, but what is common between all the approach that we can use for mapping the fluxes is that without the existence of a very well-defined variogram of the data, we cannot use this method in a reliable way. So the map that we are going to produce using our dataset, if this mapping is not based on the existence and on the modeling of the variogram of the data, is simply a picture, is not some quantitative model of the spatial distribution of the data. And because from a theoretical point of view, our dataset need to be stationary to be elaborated with this geostatistical tool. And in practical terms, we can detect and define if our dataset is stationary by looking if exists a variogram, and if the variogram increase with the distance between the points, and if this variogram reach a threshold values, and if this threshold values is similar to the variance of the data. In addition, the variogram tell us also some important features of the spatial distribution of our data. So what is the variogram? The variogram is a function that in some way is going to measure how much to measurements, much to flux values are different when are separated by a certain distance. And what we need to have is that the difference between the values increasing the distance of separation between the samples. This is something that is also quite intuitive, because if there is a structure of the anomaly in the space, if the measurements, if the values are very near one to the other, these are more similar. These are more distance one to the other, this has to be much more different. So what after computed the values of the variogram for different distance of separation between our points, what we want to have is that the values of the variogram increase with increasing the distance between the samples. Modeling this experimental variogram, we obtain also very other important information, because without going into much in detail in the theoretical aspect, the values of the variogram for a distance of 0, that is called the nugget effect, describe the contribution of the variability at small scale in our measurement. And generally what we want to have, that this component it's low as much as possible. And another parameter of the model of the variogram, that is the range, that is the values of distance after which the variogram remain quite constant. It tells us the maximum distance of correlation between the values. And in practical terms, this more or less coincide to the extent of our anomalies. So to see some general features about the different approach that we can use for the mapping, we can have a map produced by simulation like this, or a map produced by kriging. You see, these maps are for a certain way very similar. They both identify the same degassing structure into the space. But what we can tell about, for example, the comparison between the two approaches, that the kriging map, that is this one, appears much more smoothed with respect to this one, tend to filter our data, reducing the presence of extremely high values and extremely low values. And sometimes this cannot be useful in our representation. The other thing is that if we compare the variogram of our data sets, the experimental variogram, that is this black point in this variogram, together with the variogram of the maps, we see that while the maps produced with the sequential Gaussian simulation reproduce quite well the real spatial distribution of the data, the variogram of the map produced by the creasing has no more nugget effects that instead characterizes the experimental data. And in fact, we see that all the anomalies are very smoothed, and it's not possible in this map to have, for example, like you see here, these red points that mean high values in the middle of low values. So these two approaches in mapping, so simulation and kriging, are both can be both used, but generally is preferred for studying the carbon dioxide degassing. Is preferred to use an approach based on simulation. Another peculiarity of simulation is that the simulation can produce a series of alternative and equiprobable representation, that means equiprobable maps of starting from the same data, so we can have different products. So we can have maps describing reporting the average values of the flux, so we can have an average map of the degassing, and we can also draw the probability maps that are drawn considering a cutoff values that, for example, can be the upper limit threshold values for the background fluxes. So applying the computing the probability that at each cell the flux is higher than this background, we can produce a probability map that now are frequently used to map the diffuse degassing structure, because where the probability is higher represents the area where is much more, where is higher the probability of the occurrence of a deep flux. So these maps, this series of maps can also be used for estimate the total release from the area. For example, we can compute the carbon dioxide flux that is combined with each map, multiplying the estimated values for the area of the cell of the models, and we can also estimate the uncertainty, for example, in our total estimation, taking, for example, the standard deviation of the gas flux, of the total gas flux resulting from a series of maps. So we can, for example, estimate the total flux from 100 equiprobable simulation, and we can define which is the average flux and which is the standard deviation of this computed average flux that give to us an idea of the uncertainty of our estimation. So maybe I would skip this or go very, very rapid with these two slides to see if the example to not be very late. And one of the most frequent questions could be, how many measurements are necessary to have a reliable estimation of the gas release from an area? These are the results reported in this work of 2003. And here we did an exercise using different data set of gas flux from soil and taking away measurement from each data set and computing again the estimated total flux to see how much the estimation is linked to the number of measurements that are used for each area. And of course, we see that reducing the number of measurements, the estimation starts to be very different to that we have with the entire data set. So that means that in some way, the number of samples is impacting the quality of our flux estimate. But going more in deep to this, what at the end came out is that is not what is important is not the absolute number of measurements that we do in an area, but with what is much more important is the spacing between the measurements in relation to the dimension of the anomaly. So the general idea, what is it? If you have anomalies that are large, 100 meters, is not good to use a spacing between measurement of 200 meters or at least also of 100 meters, because this spacing is not enough to characterize the real spatial distribution of the data. So the reliability of an estimation of the total flux does not depend on the number of measurements, but on the measurement spacing in relation to the dimension on the investigating degassing structure. And to show an example about this, we will see the first with the first example that is the is published in a very recent paper that was about an area, the degassing area in a geothermal system. The area is Bagni San Filippo, and now we can see within this slide some things that we have seen in the theoretical part. For example, these are the probability plot of a general survey did in one area, together with the probability plot of some detailed surveys done in some subsets of this area. What is difference between these surveys? We will see both the spacing of measurement, but in this case, what is important is the season. The general surveys was done during the winter, while the detailed surveys was done during the springtime. And you see here, in all the cases, we have the combination of different population. We have background fluxes and anomalous fluxes. But if you look to the values, for example, the backgrounds, the biological contribution in the winter surveys is about 9 grams per square meter per day, while the lowest flux population during the other survey that was done during spring at time, where we have more vegetation, the soil is more active, we have a background of 22, 23 grams, 19 grams. So the first message is that the biological background can be also quite different between a season and another season. So in this case, the threshold that we can define for the biological flux is during the winter something like 25 grams, and during the spring and the summer something like 50 grams. The other things that can be learned from this example is this one. If we look to the map that is deriving from the general surveys and the structure of the data, we see that the variogram of the data referring to the general survey show a very high nugget effect and a range of about 320 meters. This very high nugget effect suggests that the small scale contribution is very high, and this probably suggests that the spacing usage for this general survey is not very good for defining the real structure of the fluxes in the area. In fact, if we go to see the variogram of the carbon dioxide flux in the detailed survey, where there was used a much small spacing, we see that the real range, so the real dimension of this anomaly is not 300 meters, but it's something about 58, 40 meters, 100 meters. So the message is that the general survey, the spacing of the measurement in the general surveys was not so appropriate to really catch the structure of the degassing in this area. And this, of course, impacts strongly on the estimation of the gas flux. For example, if we go to estimate the total degassing of carbon dioxide from the general survey, we obtain very large values, about 800 tons per day, which the greatest component represented by the deep carbon dioxide. Instead, if we go to investigate the flux from the same area using the general data set and the detailed survey, we see a very large difference. For example, taking this Palazzo area, that is one of these degassing sub areas, we have an estimated output of deep carbon dioxide of about 62 tons from the general surveys, while only 90 tons from the detailed survey. So this is a practical example that shows that using a large spacing for studying small size DDS can produce a great overestimation of the total release. And then this error in the estimate can reflect, for example, to other results, that can be obtained for using this estimation of the deep flux. For example, the objective of this work was to build a conceptual model of the area, and the total flux, for example, was used to compute the original composition of the gas entering into the system. And using the most reliable values for the estimation of carbon dioxide flux, we was able to estimate that the composition of this original gas entering to the system is very similar to that of the nearby geothermal system feeding the Amiata geothermals well. Instead, if we are going to use wrong values for this flux, we are not able to really define the true source of the degassing. And also this data on the total flux are also used to compute in some way the thermal energy of this system. And again, using not real values of the estimated carbon dioxide, you may obtain very not real estimates of the thermal energy associated to this geothermal system. So I think we are quite late, so I remind to the original paper. In this paper that was published on Scientific Report, you can find another example of the use of the study of carbon dioxide from diffuse degassing from the Solfatara system in terms of monitoring the volcanic activity. And so if you're interested, you can go to this paper, but I think that it's quite late. We have seen many things, and so I will stop here, leaving some space for questions.
Thanks, Carlo. Any questions?
Yeah, I have a question, Carlo.
Yeah.
Can you hear me? Okay.
Yeah.
We discovered some interesting microbial biofilms that live in a cave that contains volcanic gases in the lower parts and air on top of it. So microbial mats develop at the interface between the volcanic gases below, the heavy volcanic gases, which contain methane and hydrogen sulfide, and the air which is on top, which contains oxygen. So we see at this interface very rich microbial mats, very interesting microbial communities, and so on. I'm not going into details for that. But using this model, uh, which is a particular case of a cave, we can envision that if we go into a mass of rocks, let's consider an ideal situation where we have a pile of gravel and volcanic gases are seeping from below toward the surface of this pile, and oxygen is seeping through the pile downwards till they meet the volcanic gases. So in theory, we should have a situation where a stable gas chemocline is present in this pile of gravel. And at that level, if hydrogen sulfide or methane are present, then microbial communities could develop inside this pile of gravel at that certain level where the gas, where the redox interface is present. So what you talked about was measuring these gas fluxes at the surface of the pile of gravel, right? That was my understanding. Now, if we, is there any way in which we could lower a device that has many sensors on it so we can pinpoint the level where this redox interface is present in the soil or in the sediments around some fumaroles? The fumaroles are really holes where the gas comes out. But in theory, at least from what we learned from these caves, we should also have such a redox interface in the sediments next to the fumaroles. And that's where we could hope to find microbial community. The reason I'm asking this is that it has some implication for astrobiology, and I'll talk about it when I will have my presentation about it. But I'm just wondering if you guys have any idea, or if anybody has developed such an instrument that could be introduced into the soil and tell us where this interface is located in a certain area.
Oh, it's very interesting setting, and so, but I really don't know if there is such kind of instruments or approach for that was already developed for studying the problem that you face out. So of course the presence of microbes, the interaction between microbial community and fluxes from soil, it's also another very complex and interesting subject. And there are several studies that are trying to understand this correlation between microbes and deep-seated gases, also because this microbe can produce but can also consume, so that can hidden the deep gas, or can also, so, and it's a very interesting field. There are several works, but I cannot, unfortunately, I cannot answer directly to your question with giving a solution.
Thanks.
Gabriela Serrano had raised her hand.
Yes. Hi. Actually, it's not a question. It's only to say thank you to both of the speakers, because they were very clear, and I learned a lot about you guys. And I have a little question for Cal. So I'm doing my thesis in my last year of bachelor, I think it's their name. I am from Chile. And so I'm studying the CO2 flux in a geothermal area in the south of Chile. And I want to know if I can estimate the size of a possible magmatic chamber with my data. If I have two families of the data. can I estimate it or calculate it? I don't know if I can. I was clear.
Yeah.
Sorry.
If I well understood, the question was about how can have information estimate the dimension of the chamber of the magma chamber from your flux data. Is this more or less the question?
Yes. That was the. Sorry, I'm
Yeah.
nervous.
Yeah, I had not the time to show some other application on geothermal areas, for example. What can tell you the flux is the amount of carbon dioxide that is released by the deep system. So sometimes the question is, this is all magmatic carbon dioxide, or is produced at some other processes into the hydrothermal system. There is hydrothermal reaction. So it's not so trivial. So what you can do with fluxes is computing the amount of gas that is released in terms of tons per day, of quantity of mass per day. And this, of course, if you have your complex statistical distribution, you may distinguish how much of this contribution is from the deep source and how much of this contribution is from the shallow one. Then, for example, you can use this data for give a budget of the thermal energy connected to the degassing of the hydrothermal system, for example. And this based on the assumption that you have a vapor phase that is transporting the carbon dioxide from the geothermal system to the surface. Imagine you have this vapor that is water and carbon dioxide mixture. The vapor travels to the surface, and at certain depth it probably condenses, and what you see at the surface is only mainly the carbon dioxide, because the water part of this vapor flux is condensed. So there are many papers that is showing on how you can recompute the amounts of water that has condensed from the amount of carbon dioxide that is released. And this can give to you an idea of the thermal energy that is behind this process, because during the concentration that you have, that a lot of energy is lost by the fluid to the system. So in this way, this can be an approach to use the flux of carbon dioxide to estimate some thermal features of your deep system. But in terms of dimension of the magma chamber, you would you have your amount of gas that is released. You should know the amount of the concentration of the carbon dioxide that is dissolved in your magma, and you can do some calculation of the type that to release the amount of carbon dioxide that I see at the surface, how much magma need to be degassed. But even this is not it's not simple. It can be done. I don't know if I have answered to your question. The problem is also that in between your magma chamber and the surface, if I'm not wrong, you have a geothermal system. So the story is a little bit more complex, but.
Yes, I understand. It's complicated to
Yes,
do
but
that.
there is literature publication. You can also write an email. I can suggest you some papers that can be useful for your study.
Okay, I'm happy. Thank you so much.
Any other questions for Carlo? If not, thank you, Carlo, for the nice presentation and the great insights into diffuse degassing. So thank you, everybody, for attending this session. So see you next week.
Okay, see you everybody. Bye.
Thanks.